Swayam Prabha. Digital India. Educated India.
Good day everyone, welcome to the course on next generation sequencing technology, data analysis and applications. We have been discussing basic shell commands as part of the first hands on, and we will discuss few more commands today which will be useful for NGS data analysis. So, these are the topics that we will be covering today. So, we will discuss shell commands for file compression and decompression. So, the NGS data files, FASTQ files for example, they come with different compression sometimes. So, you may have to decompress those files. In addition, you will use many tools, they also come in certain compression. So, we will have to decompress them and install them. And the second topic that we will be discussing today is the search expression. Now, the files that we deal with, these are really huge files, right. So, you cannot just open them and search for specific read sequence or read ID if there is a need. So, we will have to use these command line tools which will search these files very efficiently. So, these are the keywords the gzip, tar and grep. So, these are the commands that we will encounter in today's class. So, let us start with the file compression part. So, the first file compression that we will use is called zip. So, this is a common file compression that is also present in Windows, you must be familiar with this compression. In Linux, so what do you do if we have to create a zip file using a command line, and similarly what do you do if we have to decompress file. Okay. So, we will discuss that one step by step. So, this is the command that we use. So, the first is zip, creating this zip file. So, how do you create this zip file? So, this is the command zip. So, the commands are shown in green, right, that a part of the command, and then we have the file name that you want to create after zipping. So, this is file1.zip and followed by the file name from which you want to create this zip file. So, the file 1 is the file from which we want to create this zip file. So, this is the file name that is given. Okay. So, this is a slightly different notation, right. So, the zip file name would come before the actual file name. Okay. Now, similarly, if you want to compress a folder itself containing multiple files, this is how you should write this. So, you have this, you would have to add this option zip minus r. Okay. So, minus r means recursively. So, it will compress all the files, all the contents of that folder into that folder 1.zip. Okay. And then followed by the folder name from which you want to create this folder. Okay. So, we will see this in a moment how this works. So, how do you unzip files? So, for unzipping files you just simply say unzip and file name, the zip file name from which you want to create this decompressed file. Okay. So, this is how the thing will work, right. So, let us try this out in the command line, right. So, we will try that out, and as part of the hands on you can try in parallel. Okay. So, if you remember some of the commands that we have used last time. So, one of the commands is ls, right. So, this lists file that are there in this directory. Okay. So, this is the file name D12 some specific FASTQ file, this is a FASTQ file we are working with, right. So, let us zip this file, right. So, how do we compress, right. So, the command is zip compress dot zip and then the file name, right. So, the file name is the D12, the full file name, right. This FASTQ file we want to compress into a zip file, right, and so we will call this new file as compress.zip. Okay, and this is done now, right, and you can see this zip file is there, okay, compress.zip. Okay. Now, if you want to again unzip this, right, so we simply write unzip compress.zip, right, and it will say replace. We do not want to replace, or if you want to replace you can say yes, if you do not want to replace you say no, just right. So, you just have this, right. Okay. So, this is how this zip and unzip works. Okay. So, this is a very common compression that we use also in Windows. Then you have other compressions, these are actually Linux specific compressions, okay. And these are more efficient, you can actually control the level of compression as well. Okay. So, the command the compression that we use very commonly is called gzip compression. Okay. So, you will see this in FASTQ files, sometimes they will also come with this gzip compression, and you will see this .gz at the end of the file names. Okay. So, how do you compress this is gzip file name. Now, if you do gzip file name, what will happen is that the program will simply take this file name, right, this file, and compress it, and the original file will be lost, you will not have that original file anymore. Okay. Now, if you do not want to lose that original file, these are the options that you have. Okay. You can say gzip -c file name to file name .gz, right. So, the format is slightly different from zip. Here it is you want to create this, right, gzip -c file name to file name .gz, or you can simply say gzip -k file name. Okay. So, -k would suggest I keep the original file. Okay. So, this is how this whole thing will work. The other thing is you can control the level of compression with gzip. Okay. The levels are 1 to 9. 1 is the least compressed, means it will take more space in the hard drive, and it is the fastest, right. It does not need to compress too much. So, it will be very quick, whereas 9 is the slowest and it is the most compressed, right. So, how do you actually control this, right? How do you control this level of compression? You say gzip -2 file name. Okay. So, these are the options. So, you probably getting an idea of how to use these shell commands, right. You have the commands, and following that command you can sometimes use certain options. Okay. So, this is very common across all Linux commands. Okay. In case you want to compress a folder, you have to again give gzip -r folder 1, right. This is again same with what we have seen earlier, right. If you want to compress a folder, you just say zip minus, right. So, let us try this out gzip in our command line, okay, and this is what we will do, right. Gzip, we just give the file name, okay, and this is done, right. And what you see here is this file now, original file, right, D12 this read1 part dot fastq dot gz. Okay. Maybe you can zoom in a bit, so that it's easier for you to see, right. So, let us do the ls again, and you will see this, right. This fastq file, original fastq file is now fastq.gz. Okay. So, there is this gzip compression. Okay. Now, how do you undo this, right? Right. So, okay. So, we have not talked about that yet, right. How do you actually undo this gzip, right. The other thing we talked about is if you want to create, if you want to keep the original file, right. So, first we have to undo this, right. We have to decompress. So, the decompression works like this. So, these are the two commands that we use for decompression. One is this gzip -d file name .gz, right. You give the gz file and say gzip -d, or you can simply say z unzip. Okay. So, let us try that z unzip, right, and then we will try the other commands that we have discussed with gzip. Okay. So, z zip -d, right, and the gz file. Okay. And it is done, right, and you can check, right, just by saying list you can simply check, and we are back to the original .fastq format. Okay. And now let us try this gzip -c or -k, right, keep the original file, right. So, gzip -k, right, okay, and it is done, right. And you see now there are two files, right. So, the original file, the fastq file is there, and then we have this fastq.gz, right. So, you get an idea of how these commands actually work. Okay. All right. So, let us move on to the next compression, right, that is also quite common in Linux systems, and you will see many software tools that will come with this compression. Okay. So, this is the command we use, it is called tar, right. So, what it does, it creates an archive by grouping multiple files into a single file. Okay. And it is a really elaborate command, it has lot of options and you can control a lot of things, right. So, if you want to compress multiple files, what you will do, you will take this file 1, file 2, right. Look at the format of this command, you have tar minus cf, right, archive.tar. So, the file that you want to create, that should come first, and the extension is .tar, okay. And the 2 file names or 3 file names, multiple file names, as many files as you want to compress, you just put those file names one after another. Okay. So, here in this example, we want to compress this file 1 and file 2, right. So, we create this archive.tar from file 1 and file 2. Okay. If you want to extract, okay, so if you want to create, we use this minus c, right. So, f means file, so minus cf, right. So, create this file, okay. X means extract, okay. So, that is why the option minus xf, okay. So, you have created a tar, right, and you want to extract this, okay, and you will write tar minus xf archive.tar, okay. If you want to extract these files into a specific directory, let us say you have hundreds of files, you know there will be many files in this archive, right. So, what you do is you want to specify a directory instead of the current working directory, you want to specify another directory where this extraction process will happen, right. All the files will be extracted into that directory, and this is the option you will give minus c directory, okay. Now, if you want to create a zzip version of this archive, okay. So, we have talked about this zzip, right. So, here we are creating this archive tar, on top of that we are applying this zzip compression, right. So, you will see this .tar.zzip, right. So, this is something you will see for many tools, many softwares, many sometimes many data files also, if you want to achieve a really good compression from multiple files, okay. So, this is the command you will use tar minus czf, right, archive.tar.cz, right. So, you see these 2 compressions .tar.cz. So, you know like okay, this is a compressed zzip compressed, and then the file names that you want to create the archive from, okay. Now, if you want to extract this, right, so again you can simply use tar minus xf, actually tar knows if it sees .tar.cz, it automatically knows okay, this is zzipped, okay. So, you do not have to again specify, right, that this is a zzip file. So, use this minus xzf or minus zxf, but if you want you can also use minus zxf, you are explicitly mentioning this, right, minus zxf here, okay. So, this is the these are the 2 options that you have. You actually have many more options here. So, some of them we have talked about. So, minus c is create, minus x is extract, minus f is file, right, minus z is zzip compression. So, if you want to create this zzip compressed archive, right, we use this minus z option, and there is another type of compression which is called bzip2 compression in Linux, okay. So, for that we use this minus z option, okay. And sometimes we also use this minus v option, which means the program or that command will tell us what it is actually doing, okay. It will kind of give an kind of step by step process that is going on, okay. So, let us try this, right. These are some of the examples that we have, right. You can merge these commands. So, if you are working with this tar.cz file, you want to extract, you will say tar minus zxvf, okay. This is something that will be very useful when we want to install tools. You will see many of them will come in this .tar.cz format. If you want to extract this tar.bz2 file, right, you will use this minus zxvf, okay. This is something again, if you see this bz2 compression, this is something you will have to use, okay. So, let us try this out, okay. Let us try this in our command line, and let us get a feel, right. Of course, we will not try all the commands, but at least if we try some of the options, you will get a better understanding, right. So, if you want to create this archive, right, what we have to do is first we will have to get another file, right. We have this .fastq, let us get another file, and we can utilize a command that we discussed last time, right, if you remember, right. So, if you remember, we use this pwd command, which is the present working directory, right, and inside this test 1 we were inside this test 1, right. If we go out of test 1, right, you may not remember, right, if we go out using cd dot command, there is this genome data analysis folder. Okay. So, inside this folder again there are multiple files. So, what we will do, we will copy another file, so that we can demonstrate this compression, right, creating this archive etcetera using tar. Okay. So, let us do that. So, what is the command for copying? So, it is actually cp, right. So, cp, the path name where this file is, right, this is the folder inside which we have this another file, and let us copy the other file that is there, another FASTQ file is there, and we copy this to the test 1 directory. Okay. So, this is the format we discussed last time. CP genome data analysis is the file name and the destination directory. Okay. If the destination directory is the current directory, we can simply say dot. Okay. We can just leave with dot, means so the command will understand the destination directory is the current directory. Okay. But our destination directory is test 1. So, we will move into test 1, right, and we will see like this file is there now. Okay. So, again we go inside this test 1, right, and we can check that these files are there. Okay. Now, we see these two files, two FASTQ files, okay, and what we want to do, we want to create an archive out of these FASTQ files, right. So, these two FASTQ files, let's compress them, and we use this tar command, okay, minus c, right, we want to create this compression, z, okay, we want to compress with zzip, right. So, zzip compressed and f, okay, and maybe if you want to see, right, what the program is doing, right, we want to also use this option vf, right, what the command is doing. Okay, it will tell us what it is doing. Okay. So, the command again is, you remember, right, compress tar dot cz, right. The file name we have to give the archive that we want to create first, followed by the two file names. Okay. We want to compress the FASTQ files, right, two FASTQ files. Okay. So, these two files are there. Okay. So, let's run this command, and it will tell us, right, what it's doing. Okay. So, it just gave, okay, this is reading these two files and it has created this compression. So, let us check, right, if the compression is there, if the archive is created, you see this compress tar dot cz is there. Okay. So, this has been created. We can check the sizes, right, of these compressions, and you can compare, right, how tar, how zzip and how zip actually compare in terms of storage space, and you can see which one is the most efficient, right. This is something you can try. We will see the command how you can check this a bit later. Alright. So, now we want to unzip or decompress, right, this tar dot cz. What is the command? This is tar dot cz. So, this will be tar minus zxvf, right. X is for extract, right. You can simply say xvf, right, instead of saying zxvf, but we can use this, and the file name. Okay. Right. So, let us unzip this. Maybe we want to do this in another directory, right, maybe not here. So, we can create this directory and we can say, okay, do not extract here, extract in that directory. Okay. So, let us do that first. Okay. So, how do you create directory? mkdir, right. So, we have discussed this in the last class, and we say we create this test2 inside test1 directory. Okay. So, if we say ls, you see this test2 directory has been created here, right. This is there, test2 is there. So, now let us unzip or decompress this, right. tar minus zxvf compress tar dot zz, right. How do you specify the directory? Minus c test2, right. You remember we have just seen, okay, and again it will show, okay, this has extracted these two files, and inside this test2 directory it should be there. Let us look at the files whether they are present inside this directory. So, the command we use is ls test2, okay, and you see these two files are there. All right. So, I think this kind of gives you an idea, right, how to compress files or decompress files, right, especially the decompression will be very important for you when you download tools or extract these tools, right, and before you use them you have to do this step. Okay. So, let us now go back to the presentation and we will discuss a few more commands that could be useful in many cases when you are analyzing NGS data. Okay, so, the one command that we use is called WC command. Okay. So, this gives you a lot of useful information about a file or an input. Okay. So, this is something that we can use for a variety of application, but I will talk about only two applications in this case. Okay. That will be very useful for you probably, right. So, one thing you can do without actually opening a file, you can count how many lines are there in that file. Okay. So, this WC command will tell you how many lines are there in that file. Okay. So, something called WC minus L, okay. The option is minus L and the file name. So, here the file name is file.txt, right. It could be any file that you want to check, or you can simply count the number of words in a file. Okay. So, here is this WC minus W file.txt. Okay. So, let us try this, right, in the command line and see how what you get from this outputs. Okay. Okay. So, we cleared the terminal, right, so that you can see well, and again we use the command ls, right, and you can see we have these FASTQ files, right. We will try these commands WC minus L on these FASTQ files. Okay. So, let us try this WC minus L D12 the file name, right, this is a big file name dot FASTQ, and we will run this. Okay. Okay. So, what you see is the number of lines in that file. This is, in this case, this is 100,000 and the file name. Okay. Now, we can do the same for the other file, right, D12 read 2 and see how many there are 100,000 lines, right. So, this makes sense because if you look at these file names, these are read 1, read 2, right, they come from the same data, they are paired in data, right, and contains information about two mates, right, mate 1 and mate 2. So, they should contain exactly same number of reads, okay, and exactly same number of lines. Okay. So, that is why we see these 100,000 lines in both of them. Now, let us try the other command, which is the WC minus W, right, and then the file name. The number of words is given 125,000 here, and for read 2 this is the same, this is again expected. Now, one of the things you might be thinking about, right, how am I writing these big file names so quickly? Okay. So, there is a trick in the terminal which I am going to tell you, right. So, let us say we want to run this again WC minus L, okay, and if you have a big file name, right, if you just write tab, if you press the tab button, it will show you what are the files that are there inside that directory. Okay. Of course, try this only when you are sure that you have only few files in that directory. If you have 100,000 files, you do not want to try this because it will take a long time to show you all the files. Okay. Now, if you want to see, right, what are the files that start with these letters D12. Okay. Again, so you type D12 and then you use tab. Okay. Immediately it will give you these extensions automatically. You see it will find these matches and it will automatically take this. Okay. Now, it will not go further because it finds two options, right. It has this read 1 underscore part and the other one is read 2 underscore part, right. So, now you have to specify which one you want to take, right. If you want to know what are the options that are present, you simply type press tab twice, right. Then it will tell you, okay, you have these three files that first part for which matches, right, with this expression, then you use, let us say we specify 2, okay, and then press tab again and it will take the rest of the file name. Okay. So, this is how we can type really long file names really quickly. Okay. You do not have to always type through the letters, because that is going to take a lot of time. You can simply type a part of this file name and use tab. Okay. So, this is a very useful way of writing very big file names. Okay. All right. So, you have understood, right, WC minus L and WC minus W, how they work. Now, let us go to the other important expression that is actually very useful, as I have mentioned earlier, right. So, this is called the grep. Okay. So, the full form is the global regular expression print. Okay. So, what is this global regular expression print? So, as I said, like for example, you want to find a file name, let us say a read name in your file. Okay. Or it could be, so if you are not talking about ngS data, for any file name you want to find a specific expression, specific name or specific data point, right, which has a unique name. Okay. So, for doing that we use this command grep. Okay. So, it can search this whole file very efficiently, and it can give you the instances where this has found this expression, right, this text or expression. Okay. So, this command is used as I mentioned to search for text or element in a file, right, or you can also search output of another command. Okay. This is something we will not discuss, right, but you can of course look up. So, if you utilizing another command in this Linux command line or terminal, right, you can actually pass that output of that command through grep, right, to search for specific outputs, okay, or specific patterns, right. So, this is something that can also be done, right. So, this is a very useful tool. It has a lot of ways of applying it, right. So, you will probably learn as you go along, like if you learn more about Linux. We will use this for a very simple kind of task. Okay. We want to find a specific name inside a file. Okay. So, for example, we want to find a read name inside a file, or we want to find, for example, once we have done the ngS data analysis and we have got the mutation data, we want to look for certain chromosomes, right. Maybe there are mutations in certain chromosomes that we want to look at, let us say chromosome 5 in the human genome, because that might be useful for certain disease identifications, classification, etc. Right. So, this very simple task of grep could be very useful for many applications, right. So, this is how the expression goes, right. So, grep is the command. You have this time, which means this is the text that we are searching for, and then we have this file.txt, right. This is the file where we want to look for this time. This is the expression that we want to look for in that file. Okay. Now, let us look at this, right. We will see this in a moment in the command line. There is another option that you can also take, right. For example, you want, like, you just do not want just the occurrence, you want to get also the context of this file name, right. So, context means maybe just show us the first the 2 lines before the occurrence and 2 lines after the occurrence. So, grep can also do that, right. So, this is where we use this, right. So, grep, the option is -n capital C, right. So, note these options, right, whether they are in capital or small letter, this is very important, right. These are actually critical, you cannot just interchange small and capital. Okay. So, this is something that you should note for all commands. Okay. So, if you use this -nc2, right. So, this means print the context 2 lines before and 2 lines after the match. Okay. So, you are searching for this expression time in this file, right, and wherever there is a match, the command will also print 2 lines before the match and 2 lines after the match. It kind of gives you the context, right. This is useful for some applications where you probably want the context as well. Maybe there are multiple matches, but you are probably looking for just 1 or 2 places or 2 contexts, right. So, this is something that is very useful. So, let us try this out, right, in the command line. So, what we will search for here, okay, we can search for certain read names, etc., right. So, let us search for certain read names, okay. Maybe we can search for certain number. So, let us try, okay, if we can find this, right. This number probably will occur in the header line, right. This is a FASTQ file, this number might occur in header line. So, let us see if we can find a match somewhere. Okay. So, this is the expression, right, grep 10224 D12 this the file name, okay, and we will search, okay, and it returns 2 results, right. It has found in 2 places this number 10224, right. So, you can search for anything you want using this command, and it also highlights, right, this has found this in this red color. Okay. Now, let us try the other one, right. We want some context, okay, not just the results, but also the context. Okay. So, this is again the format. We use this option minus n capital C and 2, right. We want 2 lines. If we want 3 lines, let us, in case let us say we want 3 lines, we say 3, the expression that we are looking for and the file name. Okay. And this is what you get. Okay. Maybe it is slightly cumbersome, it is difficult to read at this point, right. So, it gives you the context, also the line number. Okay. So, where it is actually finding this in the file. Okay. So, on the right in green you see the line number, right, where it is finding this, and some of the data. So, here it has found this 10224 and it is giving this 3 lines before this and 3 lines after this. Okay. And similarly, for the second match it will also give you the context. Now, you can also take this out and you can store them in another file, right. If maybe in the terminal, if you are seeing this output, it becomes very cumbersome. So, you can say, okay, store this in a temporary file temp.txt, okay, and you can simply channel this output using this greater than sign. So, it will go into this temp.txt, okay, and let us do that, and see it will not print anything here in the terminal. It will write everything in the temp.txt. Okay. Now, how do you actually get this temp.txt? We have used certain commands, right. We have, remember, right, we have discussed this. How do you get this in the terminal from a file? You can use cat, right. You can say cat temp.txt, or you can also use less, right, temp.txt. You have discussed this in the last class. Okay. And you can see, okay, this output file is now contains the output of that command line. Okay. So, this kind of helps in storing whatever you have done. Okay. So, just to summarize, so this is the reference that we have used, and to summarize, we have discussed these shell commands that allows us to navigate through Unix systems, as well as the data compression and decompression commands. Okay. And these commands are really useful for many tools, softwares that we will use for NGS data analysis. Thank you.